Welcome grade 12. I want to welcome you to the Junior Tukkie Winter School. There will be a series of videos which will be live streamed in the next few weeks on the juniortukkie.online platform. Please use the given material as revision and also look at all the grade 12 content for life sciences on our platform. So let's get started. I am Ms. Kutsia, and today we will look at the Life Sciences Paper 2 that was written in November 2021.
Welcome back grade 12s. Today we will look at question 2. So question 2 will form part of section B, where section B is a total of 100 marks out of the 150. So the first question we will look at and deal with is question 2.1. So again, before you read any of the questions, you have to analyze and examine the diagram that's been given. So please remember that it's so important to read that first question because that's your first clue to know what this question is about. So let's read. It says, the diagram below represents two processes that occurs during protein synthesis. So what this tells me is that I am going to make a protein because the word synthesis means I am making something. So at this point, I'm going to make a protein. If I look at my diagram, I will see that I have DNA. Then I have process X that takes place where it converts DNA into mRNA. And then we have process Y that takes place, which converts my mRNA into a complete protein, or like in the diagram, a peptide chain. So it is so important for you to understand that X in this case is going to be transcription, and then Y is going to be translation. Then this will solve all your problems because this is going to help you to answer all the questions that follows. So let's quickly have a look at a summary of what is protein synthesis. So on my left hand side, I have three types of RNA. So the first one that we won't work with is rRNA. So this is ribosomal RNA. Then we will definitely work with mRNA, so this is my messenger RNA. And then lastly, the one that we will also work with is going to be tRNA. So this is your transfer RNA. Then if we move to the right-hand side to my second picture, we will look at what happens during protein synthesis. So firstly, my DNA unwinds and it unzips. RNA polymerase will make a template or complementary strand of mRNA, and then the mRNA will move to the ribosome in the cytoplasm, and tRNA will then form my peptide chain. So this is just protein synthesis in short. You can go back to the content that's been loaded up for grade 12s, or you can even go back to older question papers where they give you the exact marked allocation in the marking guidelines, what they expect from you. So it will tell you exactly where the mark allocation is given, and then obviously exactly what is expected from you to know for protein synthesis, because this topic actually can get quite difficult. So let's move on to reading our questions. So firstly, again, we start with the first sentence, which is very, very important. So firstly, they say that the diagram below represents two processes that occurs during protein synthesis. Then my first question asks, where in the cell does each of the following processes occur? So the two processes that they refer to is process X and process Y. So the most important thing here is that you need to be able to identify these processes. If you do not know the name of the process, you will not be able to answer the location, or you will not know where it will take place. So process X will be transcription and process Y is translation. Question 2.1.2 states one difference between the type of nitrogen bases found in DNA and RNA. So here again, you need to know what a nitrogen base is for you to be able to answer these questions. And they asked for one difference between the two. They did not state that you have to put this in a table, so you can simply just write it in the form of a sentence. Question 2.1.3, they ask you to name and describe process X. So this was quite an easy one. Usually they will give you a picture of transcription and ask you to discuss or explain translation or vice versa. So this should be an easy 6 marks, as you can still answer this question even if you don't understand it, because you just have to memorize the memo. So, but very important, there will be a compulsory mark given to the name of the process. So the name will firstly for X will be transcription, and that 1 mark is compulsory. Then to describe the process, there will be 5 marks left for you to describe the process X that takes place in protein synthesis. So let's look at the marking guidelines, so where the marks are allocated. So question 2.1.1, they just wanted the location where these processes take place. So transcription takes place in the nucleus, or you can also say the nucleoplasm, and then transcription will take place at the ribosomes, or specifically in the cytoplasm. Then question 2.1.2 wanted you to give one difference between the type of nitrogen bases in DNA and RNA. So here you can write it in 2 ways. So you get 2 marks, and I only mark the first one. So you can state and say that DNA contains thymine, which is 1 mark, whereas RNA contains uracil. That's your second mark. And you must always remember that you cannot answer in the negative. So you cannot answer and say DNA contains thymine, whereas RNA does not contain thymine. You will not be rewarded the mark. So you have to be very specific and say that RNA contains uracil. You will also notice that even if you just wrote the letter for the nitrogen bases, T and U, the mark was also rewarded, and we will mark the first ones. Then question 2.1.3 said that you had to name and discuss the process. So a compulsory mark was given for the name of the process, and process X is known as transcription. And then there's an extra 5 marks for you to describe the process that's taking place. And as you can see, there is about 1, 2, 3, 4, 5, 6, 7, 8 possible marks that you can be rewarded when describing this process, and you only need 5. So firstly, your double helix will unwind, and it will then unzip. When it unzips, it means that the weak hydrogen bonds are being broken. So instead of writing unzip, you can also say that the weak hydrogen bonds between the nitrogen base pairs are breaking. So this will form then 2 separate strands. So this is very similar to DNA replication, but now the difference comes in is that only 1 strand will act as the template, where in DNA replication, both strands act as a template. So the 1 strand that acts as a template will be used to form mRNA. And how I make the mRNA is I use a protein or an enzyme that's called RNA polymerase, which then adds free RNA nucleotides from the nucleoplasm to make the complementary strand of mRNA. So in this case, I'm not going to use DNA nucleotides. I'm using RNA nucleotides because I'm making mRNA. And from there, my mRNA, which then codes for the message of the protein, will then leave the nucleus and then enter the cytoplasm. So question 2.2 is just one single question. It asks you to describe how non-disjunction may lead to Down syndrome for 5 marks. So here you cannot name, it has to be described. Here I've added a few photos to give you an example of what Down syndrome is. So this is a genetic disorder that occurs on chromosome pair 21. It's also known as trisomy 21. So here's a small summary that shows you what happens. This is specifically non-disjunction that occurs in anaphase 1. So your chromosomes will line up on the equator and then they fail to separate. So the homologous chromosomes will move as one to the one gamete when meiosis 1 takes place. So something similar will happen during anaphase 2, but here the chromatids will fail to separate and there will be a complete chromosome moving to the opposite pole. So let's have a look at my reading my question and also my marking guidelines. So we have to describe non-disjunction that leads to Down syndrome. So firstly, this will happen when chromosome pair 21 fail to separate during anaphase. So here you can see you do not have to be specific to say that it's in anaphase 1 or 2. You can even say this occurs during anaphase 1 and 2. So you can mention it, but the important part is anaphase. So what happens is that a daughter cell or gamete will have 24 chromosomes instead of the normal 23, or you can say that the gamete will have an extra chromosome. So when this gamete is then fertilized with a normal gamete with 23 chromosomes, it will then form a zygote with 47 chromosomes instead of 46. So, or you can also say that there's 3 chromosomes that's extra, or there's 3 chromosomes at position number 21 instead of just 2. So this is also then known as trisomy 21. So here you will see a diagram that represents the karyotype of someone that has Down syndrome or trisomy 21. So if you move down to the number 21, so that's position number 21, and there you will clearly see that there's 3 chromosomes, which will represent obviously Down syndrome, and instead of your normal 2 chromosomes. So next we have question 2.3. So what you are looking at in these 2 diagrams is a karyotype. So they specifically state here that the diagram below represents the chromosomes from a human somatic cell. So that means it is a normal body cell with 46 chromosomes of 2 individuals who are twins. So at this stage you are not sure if they are identical or non-identical twins, but if you know how to read a karyotype, it is quite easy to distinguish if they are identical twins or not. So you have individual 1, they give you the chromosome pairs from 1 to 23, and the same for individual 2. So let's quickly have a look at how to read a human karyotype with 46 chromosomes. So in position number 1, we will actually have 2 homologous chromosome pairs. So there's 23 pairs of chromosomes in any human somatic cell. So definitely not a gamete, because there we will only find 23 chromosomes. So from pair, okay, so we have a total of 46 chromosomes or 23 pairs. The first 22 pairs, so from pair number 1 to 22, we call them autosomes. And then the last 2 pairs of chromosomes, which is pair 23, we call them gonosomes, because these are your sex chromosomes. These 2 chromosomes will determine the sex. If you are male, you will have an X and a Y chromosome. So this will be represented by a longer chromosome and a short chromosome, like in the picture. If you are female, you have 2 X chromosomes, meaning that both of the chromosomes will be equal in size. So if you look at this picture where we have X and Y, we will, for example, then have 2 purple chromosomes like in the picture if this would have been a female. So let's read the question and what is asked from us. So again, these are the karyotypes or the chromosomes of a somatic cell of 2 individuals that are twins. So the first question is, what is a somatic cell? There's nothing tricky towards this. If you did not study it, you will not know what it is. Then they want you to name the specific type of chromosomes that's numbered from number 1 to number 22. Then each of the pairs shown in the homologous pair of chromosomes. State the origin of each chromosome in a homologous pair during the zygote formation. So they actually want to know, if I look at one pair, I have 2 chromosomes. So where do I find these 2 chromosomes? Where do they come from as soon as I form the zygote after fertilization? Then they want us to list 3 characteristics that chromosomes in a homologous pair have in common. And then you have to explain one observable reason why the 2 individuals are not twins. So you have to be able to see it in the given diagrams, in the karyotypes. So let's look at the marking guidelines. So firstly, what is a somatic cell? So it's a simple body cell. So it is a cell that you find anywhere in the body except for your sex cells. Then name the specific type of chromosomes numbered 1 to 22. They are known as autosomes. Then question 2.3.3, each of the pairs shown is a homologous pair of chromosomes. State the origin. So where do these two chromosomes come from? So one chromosome will come from the sperm cell or from the father, and the other one will come from the ovum, or also known from the mother. Then B, list 3 characteristics that chromosomes in a homologous pair have in common. They will have the same shape, the same size or length, the position of the genes, so the alleles, the locus where I find the alleles will be the same, and then genes coding for the same characteristics, and then also the location of the centromeres. Again, I will only mark the first 3. Question 2.3.4. Explain one observable reason why these two individuals are not identical twins. So if we do have a look at it, you will see that the gonosomes are not identical. So meaning the chromosomes at pair 23 are not the same because individual 1 has a X and a Y gonosome or a chromosome, where individual 2 has 2 X chromosomes, so or gonosomes. It's very important that you do not just say that individual 1 has a X and a Y and individual 2 has a X and a X. You will not receive the marks. So you have to state that they are gonosomes or chromosomes, and one is female and one is male. That is how we know that they are male or female. So if you go back to question 2.3.3, you'll see I added 2 homologous chromosomes. I just made them 2 different colors. So for example, let's say the blue comes from the dad and the purple from the mom. You will see that the centromeres, the location is the same. The locus of the alleles are the same. They will carry the same genes. It's the same length and the size, and the shape is also exactly the same.
Question 2.4 is about a pedigree diagram. So it is very, very important for you to understand how a pedigree diagram works before you analyze and look at what this question is about. So in this case, they say that this scenario, this pedigree diagram, is about one type of deafness in humans, and this is carried by a single allele. So the diagram then shows us how deafness has been inherited in a family. So before we read the questions, let's look at the summary of a pedigree diagram. So this is not very complex. It's something that you have to remember, is that a pedigree diagram does not necessarily mean that it is a sex-linked genetic disorder. It does represent male and female, but you need to pay careful attention here. There's only in 2 incidences where it will be sex-linked. That's with hemophilia or color blindness. Otherwise, I have to state to you if it is a sex-linked diagram. But in this case, it was just deafness and nothing was said about sex-linked. So let's just have a look at what this means. So if you have a look over here, you will see that we have a square and a circle that is connected to one another by means of a line. So this means that these 2 individuals are married. Then you will see a long line with 3 lines moving downwards. The individuals are not connected directly or next to each other. This means that these individuals are siblings. They are related to one another. So they are siblings. Then also we have the first generation, then we have our second generation and our third generation. Here is the key to the pedigree diagram. So I have seen cases where they haven't given the key before, but you will notice that the square is a male and the circle is a female. As soon as the image is colored in, so you have a black square, it means that the male has the genetic disorder, and if you have a colored in or a shaded or a black circle, you know that you have an affected female. So let's examine the pedigree diagram that's given. So they say that there's only one type of deafness that we are examining that we find in humans. So this is caused by a single allele that's carried by an individual. So here they show you in the pedigree diagram how this is inherited from one person to the next. So let's quickly have a look at my image, as it is extremely important to examine my pedigree diagram or any given diagram. So firstly, we have Paul and Lizzie, which forms part of our first generation. So Paul and Lizzie had 3 offspring, 2 unaffected boys and 1 affected female. And then we have Mary and John, which is also part of their first generation, which then had 2 offspring, which is Anne, which is unaffected, and then also a boy that's unaffected. And these individuals form part of the second generation. Then from the 2 families, Bob and Anne got married and they had 3 offsprings, where you will notice that they have 1 female that's unaffected, 1 male that's unaffected, and then 1 male that was affected. So that means that this male carried the gene for deafness, or was actually affected by the allele. So they will form part of my third generation. So how many generations are represented in the pedigree diagram? So if you go back to my summary, there I have indicated how one can find the generations of the pedigree diagram. So here you will see that Paul and Lizzie, Mary and John will form the first generation. Bob and Anne will form the second generation, and their offspring will form the third generation. Then the next question is children of Paul. So how many children of Paul and Lizzie are able to hear? So here you just have to examine their children and not the entire pedigree diagram. So you have to look at the second generation, Bob and his 2 siblings. Then they ask which phenotype is dominant. So here you will have to look at how the gene or the allele is inherited by the offspring for you to determine which allele is dominant. And this can be quite tricky. Question 2.4.3, they ask you to use the offspring of Bob and Anne to explain your answer to question 2.4.2. So you have to use their 3 children. They had 1 daughter and 2 sons, 2 affected and 1 unaffected, and obviously both parents that was unaffected. So this can already give you an indication of or if the gene, which gene is dominant. Is deafness dominant or recessive? So this is very important to make use of Bob and Anne to explain your answer to why the allele for deafness is dominant or recessive. Then question 2.4.4, they ask you to use the letter A, so it's a capital letter A, to represent the dominant allele, and then a lowercase a to represent the recessive allele. And then you need to choose then a genotype for the individuals that has the ability to produce all the possible genotypes for someone that will be able to hear. So this is very, very important for you to go through. This can be quite tricky. They just actually want the genotypes. There's no need for you to do a genetic cross. You won't get marks for it, but it will be very easy for you to do a genetic cross to help you understand or how to get to the answer. So let's have a look at what the marking guidelines gives us. So how many generations are represented in the pedigree diagram? So according to the memo, there's 3. So let's have a look. So Paul, Lizzie, Mary and John forms part of generation 1. Bob and Anne and their siblings forms part of generation 2. Just note that Bob and Anne are married to one another and they are not related to one another. And then their offspring will then form part of generation 3. Then they want to know how many children of Paul and Lizzie are able to hear. So these will be indicated by a a square or a circle that's not colored in. So that means they are unaffected and they are normal. They can hear. So here you can clearly see in the picture that we have Bob and then his brother. So there's a total of 2 offspring or children that will be able to hear normally. Then question 2.4.2, they ask us which phenotype is dominant. So that means the one that will mask the other allele, and that would be hearing. So to figure out that hearing is the dominant gene, I actually quite feel like it is quite a difficult question. It's a difficult question to answer. So usually what I do to make it more easier for me is that the most prominent allele that you will find in the pedigree diagram will be the dominant allele. So in this case, you will see that there's about 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 people in this pedigree diagram where only 4 of them has inherited the allele for deafness, where then the rest, about 8 of the other people of the 12, has normal hearing. So that's how that's how it explains to me which of the alleles are dominant. But also again, if you do have 2 individuals, let's say for example Bob and Anne, these are the 2 easiest explanations for me to use in generation 2 to explain if something is dominant or recessive. So Bob and Anne are both normal hearing individuals, and they produce 2 normal offsprings and 1 affected offspring. So that means that the affected offspring had to inherit a recessive allele from Bob and also a recessive allele from Anne. The reason why Bob and Anne carries a recessive allele for the deafness is because Anne had a mother that had the allele for deafness, and Bob had an allele, had a father that had an allele for deafness. So that means Bob and Anne were both heterozygous. And for heterozygous, they were normal hearing. So when you are heterozygous and you are normal hearing individuals, it means that you have the dominant gene that masks the recessive gene. And in this case, the hearing gene masked the recessive gene that was deafness. Then question 2.4.3, they say, use the offspring of Bob and Anne to explain your answer to question 2.4.2. So you cannot just use any individuals given in the pedigree diagram. They specifically requested that you use Bob and Anne. So firstly, you start off saying that Bob and Anne can both hear. They're normal, they can hear normally. Then if we look at their children, we will see that they have a child. They only have one single child that is deaf. So this tells you that both of the parents are carriers of the deafness allele. So the parents were heterozygous because they produced 2 normal children, and then they produced 1 child that has deafness. So that means there was an allele from Bob. So a small case, if you read question 2.4.4, a small or lowercase a that was received or transferred to their son, and also transferred a lowercase a or a recessive allele for deafness to their son, which is part of the 3rd generation. Then question 2.4.4. They say use the letter capital A to represent the dominant allele, and then the letter A for the recessive allele to give all the possible genotypes for a hearing individual. So all you need to do is you need to write down the genotype for people that will be able to hear. And if you have a look over here, so it's important to remember that hearing is dominant. So if someone is homozygous for hearing, it will be a capital letter A and a capital letter A. If someone can still hear but they are heterozygous For hearing, it means that they have a capital letter A and a lowercase a because they are heterozygous. So question 2.5 has to do with a genetic cross. So this is quite an easy question to answer if you know the format and if you know how to do it. So let's quickly read through the question as well so that we can see what is asked from us. So they say use a genetic cross to show how gender in human offspring is determined by the sex chromosomes of the parents. So a summary of a genetic cross or how this is done. So firstly, you can write your phenotype, which is male and female. So remember, the phenotype is what you see. Then the genotype will be an X and a Y chromosome for male, which is crossed with two X chromosomes, which represents female. Then meiosis will take place to form gametes. And in each gamete, I will have an X. And then in the next gamete, I can have a Y. And then in two of the other gametes, I can have two X chromosomes. To form offspring, we have fertilization that takes place to form a zygote. And then we have the Punnett square. So what happens is you link the column with the corresponding row. So we take the X from the column and the X from the row. We take the X from the column at the top and we take the X from the row on my left-hand side. We take the Y from the row and then the X from the column. And then we do exactly the same. So if you look at the Punnett square, you will see that you have exactly a 50% chance of being male and female. So then you have F1, which is your first generation. You write the phenotypes. You have two males and you have two females. And then the genotype, you have two XY chromosomes and two X chromosomes. And that it will be the representation of your format of the genetic cross and how to get the gender of the offspring. Thank you, grade 12s. Please make sure to watch the next few videos on the question papers.